Good day, everyone, and welcome to the second installment of our international workshop on HBV Cure, held virtually this year. And next slide. On behalf, I'm Adam Gehring, and a co-organizer from the Toronto Center for Liver Disease, and on behalf of my co-chair, Harry Jansen, I'd like to welcome everybody. Next slide. So the objectives for the second half of our HBV Cure workshop are really to discuss how to reach HBV functional cure and focusing on the newly developed agents targeting the immune system and the viral replication cycle, really develop those endpoints for better biomarkers and discuss how these new agents should be combined. Next slide. So the learning objectives is really to outline the progress towards HBV functional cure with these newly developed agents and understand how these new agents should be combined to reach a functional cure. Next slide. And so it's important to have partners in this throughout this process, and we're greatly appreciative to all those who partnered with the workshop and endorsed the program. Next slide. And of course, the workshop wouldn't be possible without the generous support of our corporate sponsors, allowing us to bring this to you each year. So, thank you very much. And so for those of you viewing on the live stream, you'll have your question and answer session field. You'll see it on the right-hand side of your screen. You'll be able to type the questions for the panel discussion at the end of the session. And so today I'd like to welcome our session chairs, which are Mala Maini from the University College London and Nora Turow from the Keck School of Medicine at the University of Southern California. So with that, I'd like to turn it over to Mala to introduce our first speaker. Thank you, Mala.
Thank you, Adam. It's great to be taking part in this workshop. Welcome, everybody. And I'd like to start us off with our first speaker. I'm really pleased to introduce a fellow Londoner, Kosh Agrawal, who is a transplant physician and consultant hepatologist at King's College in London, has led many trials on HBV therapeutics. And today he's going to give us an update on the capsid inhibitors. And so over to you, Kosh. We look forward to hearing this.
Thank you very much for the invitation to speak at this important international workshop on hepatitis B. I'd like to thank Harry and the faculty for inviting me to speak on what is a quite challenging topic in 15 minutes and to focus on new antiviral targets, core capsid inhibitors, and to give you an overview of the data. I'm deliberately going to focus on the data we saw from the EASL meeting earlier this year, as I felt that that is more formed, and I think that will generate more discussion. These are my disclosures. I very much feel that we need to be a little bit more provocative with regard to the data that we're seeing, and I like this quote from Walter Lippmann, which tells us when everyone thinks alike, no one thinks very much. And I'd like to thank those who provided me the slides at short notice as I skip through these slides and hopefully highlight the key issues with the data that we're seeing. Capsid assembly modulators are now really important in the field of cure for hepatitis B. They're probably the most established mode of action of therapy along with siRNA, and in essence, everybody's got one at this moment in time. In this slide, from a very nice review from MFUN and colleagues earlier this year, we see those drugs that are in red that are now in clinical trials. And also we see both those in class 1 and class 2, which demonstrate the different modes of action that we think about with regard to core protein allosteric modulators. In class 1 CPAMs, we see the formation of aberrant nucleus capsids, and in class 2, we see empty nucleocapsids being formed, and there may be some differences with regard to the function and certainly the impact this may have in the context of cccDNA influence or amplification. How do core inhibitors work. They work in several ways, and several active discussions are going on around the impact of these various mechanisms. They essentially target multiple steps of the HBV life cycle to suppress HBV DNA, pregenomic RNA, and cccDNA, and clearly cccDNA influence and diminishment is going to be critical in the world of potentially functional cure. They act here in pgRNA encapsulation to influence and decrease the formation of RNA-containing particles, to also disrupt and cause disassembly, and likewise, lastly, cccDNA amplification. And this, I think, is going to be the key as we see longer-term data as to the longer-term effects and influence on CCC amplification and diminishing that amplification as we see in various phases of hepatitis B. This is a stereotypical output that gives us a sense of the decline that we see in HBV DNA in a phase 1 study from our colleagues at Roche. This was presented last year in EASL and tells us and shows us that decrease in log over a short period of time can be fairly impressive, profound, but there is an associated rebound. This is important, but actually our interest now is looking at the newer markers as we start to understand what I've just talked about with regard to the relative effects on different components of the HBV life cycle as we understand what CPAMs can actually deliver. I'm moving fairly quickly on to the Assembly pipeline, and Assembly as a company seem to have really doubled down on core assembly modulators. This is Vabecavir or 0731. These are complicated studies, so I'm going to ask you to focus on the red squares. This is a study presented from EASL by my colleague Scott Fung, looking at e-antigen negative hepatitis B patients, comparing placebo and nuke therapy with Vabecavir and nuke therapy, and then a long extension study, which I think is relevant and important as we see longer term data. And here you can see the addition of Vabecavir to nuke therapy certainly increases HBV suppression at a very high level with regard to a very sensitive assay. And likewise, the addition over time of Vabecavir and nukes increases the suppression of DNA. Moving across to the other panels, we can see that looking at pre-gRNA and using a composite output of DNA and pregenomic RNA, we see that there is good suppression and an additive suppression with capsids in the context of nuke therapy in e-antigen negative patients. Across time, we see deeper suppression and decrease in correlated antigen and surface-related antigen. And this clearly shows us the proof of concept of adding CPAMs to existing therapy. It probably delivers more profound viral suppression and additional suppression of pgRNA. And the critical question is what is the longer-term clinical significance of this with some of the other biomarkers we're utilizing, and what might this translate to clinically? And that is really important for us as we assess this area of cure therapy. And lastly, the fairly safe review here with only one patient discontinuing due to rash. Flipping over to e-antigen positive patients, and a theme of many of these presentations is the small cohorts and the different populations. This is again from MF, who's really dominating the field of cure, and these newer commercial studies looking at not only treatment naive e-antigen positive patients, but also virally suppressed patients. Again, this is important looking at the longer term follow-up and the open label follow-up. And you can see here, both with HBV DNA and also pregenomic RNA, comparing the gray lines to the red lines. The gray is just nuke therapy alone. The red line is with Vabecavir, the 0731. You can see that there is a steeper decline and a deeper suppression of HBV DNA. And likewise, we're seeing that with the addition up front of 0731, and also when it is added at week 24. You can see here also the duration of follow-up, which is again critical as we see very short-term data generally in all modes of actions and classes of drugs in hepatitis B at this moment in time. In this presentation, what is important is that with e-antigen positive patients, 40% over time achieved a 0.5 log decline in surface antigen. This is not stunning and impressive compared to other modes of actions of drugs, but it is worth mentioning that this is more obvious in the e antigen positive cohorts that have been studied so far. And likewise, Assembly were aggressive with regard to this cohort. It's a small cohort. They followed this cohort out for a long period of time, utilizing this composite marker of DNA and pregenomic RNA at a fairly low level, seeing that up to 3/4 of patients at 72 weeks were negative at both these markers, and then were fairly brave in undertaking an assessment of whether they could achieve a quote-unquote sustained virological response. In this study, we see a press release early looking at the relapse rate associated with the 211 study, with patients followed up between 12 and 18 months. And unfortunately, what this experiment, for want of a better phrase, demonstrates is that all but 2 of the patients relapsed. So we see from this first generation capsid that this will be not enough to achieve control of surface antigen or even relapse or control of HBV DNA. Moving now to the Janssen study, somebody called Harry Janssen presented data from EASL looking at the J&J 6379 CAM. This is a group 2 class 2 drug, and this is the JADE study looking at 24-week data. Complicated study looking at not only e-antigen negative and positive patients, both not treated and virologically suppressed, but also looking at 2 different doses of their capsid. The numbers are not huge, but they are better than what we're seeing in some of the phase 1 studies. And again, what we're seeing is with both the doses, a suppression over and above nuke therapy alone in not treated patients and also in virologically suppressed patients. That's both e-antigen positive and e-antigen negative patients. I'm going to shout out here for the demographics of this population, and like many of the early studies we're seeing, this had a balance of Pan-Pacific Asian and Caucasian patients, and I'm just making the plea that we need to have studies that have the global representation of hepatitis B patients in the early studies so we can understand the heterogeneity of hepatitis B. And I think this remains a work in progress and a topic for discussion. Hepatitis B is a global disease, and we need to recognize that in these therapeutic studies. Going back to the data, we can see that surface antigen in the e-antigen positive non-treated patients decreased a reasonable amount in the higher dose, but really did not seem to move at all in the virologically suppressed e-antigen positive patients and the e-antigen negative patients across the board. So in summary, this is an important work that was presented as an oral presentation in EASL, demonstrating that overall safety was good and that the 250 milligram dose had an additive effect on HBV DNA reduction in e-antigen positive non-treated patients. Important to highlight that there were some viral ALT flares, and I think you need characterization and how community evaluate topic for discussion. And importantly, we also highlight here in this presentation, and again it's across the field with these agents, that there is breakthrough and resistance via selection if you use capsids as monotherapy, and by and large this is with the T33N resistance associated mutation, and therefore it's unlikely that capsids are ever going to be suitable as monotherapy. They're going to be used with a backbone of nuke and or in combination. To give you the breadth of what we're seeing now in the field, again, this is data looking at another class 1 CPAM. I've showed you a class 1, 2 class 2s, and now another class 1. This is a largish study given the field at this moment in time, coming out of China, looking at 110 patients treated for 24 weeks. All of these patients are e-antigen positive, and again, a cohort of treatment naive and virologically suppressed. And again, looking at the red highlighted area, you can see that in both treatment naive patients and virologically suppressed patients, you're getting an additive suppression in DNA and pregenomic RNA. It's important also to highlight in the e antigen positive population, those who are treatment naive seem to have a greater chance of achieving a 0.5 log decline compared to those who are on entecavir alone. So almost 50% of these patients achieved a 0.5 log at 24 weeks. Again, showing us that over a longer period of time that we may be seeing a secondary effect by diminishing cccDNA amplification and therefore seeing other effects that may provide a more formed, clinically significant outcome with the use of these agents. But we just don't know at this moment in time. This is merely to highlight the Alagos data. This is in preclinical models, but demonstrating that we're going to see more CAMs coming into clinical trials. And this seems to be in vitro to be quite a potent new capsid assembly modulator that will be coming into clinical trials in Q1 of next year. And we certainly look forward to being involved in that. This is another study now looking at a second generation capsid and looking at the assembly data showing again 14 days and a log and DNA change of between 2.5 in a small group of patients, 7 in each cohort, looking at various doses, and we can see the 300 milligram dose will be taken forward. And this doesn't project well, but this demonstrates that certainly Assembly have looked very hard at HBV core inhibitors and demonstrating that amino acid substitutions at the binding pocket can demonstrate differential changes in susceptibility and show us that here entecavir 0731, 2158, and then 3733 also then increase in potency. And likewise, we see better coverage with regard to the nominated resistance-associated mutations that we may come across in clinical practice. So this is an increasingly exciting field with nuanced new core inhibitors coming into clinical practice at a very rapid pace. So because Harry's now based in Toronto, I thought this quote would be useful. This is from Wayne Gretzky, telling us that pressure is a privilege in the context of the development of all drugs in hepatitis B with regard to functional cure, but particularly I'd say it's very competitive in the field of capsids at this moment in time. So a good hockey player plays where the puck is, a great hockey player plays to where the puck is going to be. And I might suggest that in hepatitis B, we're not even sure which puck we're using at this moment in time if we're thinking about functional cure. But enough of that. I'm really highlighting here, and it's for others and Marion to talk to, the importance of CPAMs and capsids and core inhibitors in the context of combination. And I'm just highlighting one study that's ongoing with Roche. There are many, many other combination studies going on. And in this study, they're using the CPAM in conjunction with a TLR7 agonist. So there's no doubt that the data we're seeing with regard to CPAMs has now meant that they're established in the field and will be a backbone in the combination therapies that we'll be using with regard to consideration of functional cure. So to summarize my talk and a roller coaster of 15 minutes or so on CPAMs, we are still learning a lot about this class of drug and certainly its effect on surface antigen in different types of hepatitis B. That gives us a hint that there may be modulation via an effect on CCC amplification, and longer term studies will give us a little bit more of a feel for what that clinically is relevant. We have a lot of promise and now a lot of early data in this field with multiple players coming into clinical trials and generating data. And they certainly seem to add something and deepen suppression in addition to nuke therapy. And that is certainly a step forward in the field with this class of drugs. I would highlight again that one size may not fit all, and we saw a slightly differential effect with regard to some of the biomarkers, with regard to e antigen status and certainly treatment status. And I would highlight the heterogeneity of hepatitis B and the need for us to have bigger data sets and longer term data sets to, number one, assess the longer term effect of this effect on cccDNA and really to understand the differential effects on different phases of disease. It's clear that this will not be a monotherapy option. There is viral breakthrough with monotherapy, and this will be a combination either with a nuke for long-term suppression, as one company is certainly taking forward as a route to licensing, or likewise as a combination with many of the other mode of action drugs that we're going to learn about in the subsequent presentations and hopefully in the discussion. And lastly, the safety profile is moderately established with regard to this mode of action of drugs, although I'd highlight that vigilance as we go out to longer term follow-up is always going to be important, and this is really a relative statement. So there's no doubt that learning and understanding the study data that we're seeing really gives us some simplicities to take forward for the field as a whole in how we manage flares, how we manage discontinuation, and really the level of suppression and the biomarkers we want to use to try and give us insights into what will achieve functional cure. So at that, I will finish and thank you for my attention and hand back to the chair. Thank you.
Great, thank you. Well, that was a terrific talk, Kosh, and thank you very much for, it's a lot to cover in 15 minutes, so yeah, hats off to you for doing so. I'm just gonna ask you one question, if I could. You've covered sort of the range of CAMs, and I guess what I'd like to get your input on is, do you think there's a fundamental difference between sort of class 1 versus class 2, either in terms of efficacy or in terms of safety? Is there any signals that you're seeing among the two classes?
So thank you very much, Nora. I think it's a really good question. If you noted through my presentation, I used a variety of different terminology to describe capsids, CAMs, CPAMs, and I think we do need to get some consensus, first of all, about the nomenclature and then the classification. And I know that the HBV Forum and other groups are starting to think about how we standardize that. You asked then a second question about, really, do we think there's going to be differential effects? I'm not so sure we're completely confident that there are differential effects, although conceptually you can see that the formation of aberrant capsids in class 1 versus empty but formed capsids in class 2 may give you some differences down the line. Maybe perhaps with regard to toxicity or even the immunological potential risk, or actually importantly, what modification of the cccDNA amplification and other processes is influenced depending on the class. And I think that, to me, is the most interesting part of this class of drugs. Will be, as we go out longer, what are we seeing is really the modification of cccDNA amplification. I don't have the answer to that, and of course it's a good question, but thank you.
Very good. Thank you very much, Kosh. That was really an excellent talk. It's now my pleasure to introduce the second speaker for our symposium today, Professor Man-Young sorry, who's from the University of Hong Kong. He really does not need introduction, because if there's a trial being done in hepatitis B with new agents, he's on it. He's leading many of the new trials, and so really is one of at the forefront of this whole entire field. He's the chair of the gastroenterology and hepatology at the University of Hong Kong. And we are really delighted that he is here today, and he's going to be given the very simple task of providing an update on new antiviral agents, non-capsid inhibitors, so sort of turning to the other side of the coin, as it were. So welcome, Professor Jung.
First of all, I would like to thank the organizing committee for this kind invitation to talk on this topic. Update on new antiviral targets, excluding capsid inhibitors. Here are my disclosures. Since HBV replication goes through different intermediate steps, it provides us different chances to suppress the virus by direct agents acting against these steps specifically. There are several possible sites of action. First, inhibition of viral entry to the hepatocytes. Second, we can lock down the RNA transcriptional activities. We can also interfere core protein functions to affect the viral encapsulation and cccDNA replenishment, and this has already been excellently covered by Professor Kosh Agrawal. Finally, we can consider to inhibit surface antigen release from the cells. In the coming 15 minutes or so, I would mention drugs for viral entry inhibition, viral RNA lockdown, and also surface antigen release inhibition from the hepatocytes. It has been shown that there are agents which can compete with hepatitis B virus for binding the anti-CB receptors on the hepatocytes. The trial agent is called Macudex B or bulevirtide. The bottom left figure showed the 24 weeks of bulevirtide had no effect on surface antigen. As shown in the bottom middle and right figures, adding bulevirtide to interferon has no additional surface antigen reductive effects when compared to interferon alone. However, the effect of bulevirtide was further studied by combining the original cohorts and new cohorts with extension to 48 weeks, followed up for until 72 weeks. It was found that there was no meaningful surface antigen reduction in either bulevirtide monotherapy arm or combination with tenofovir. However, there were more patients achieving greater than 1 log reduction of surface antigen in bulevirtide combining with pegylated interferons compared to pegylated interferon monotherapy group. Paradoxically, the best results were seen in the lowest dosing group using 2 mg bulevirtide. In this particular group, 27% lost the surface antigen at week 72. A more promising new agent or new therapy, in my opinion, is viral RNA lockdown by using either sRNA or ASO illustrated in green and yellow boxes respectively. As illustrated in the bottom right diagram, sRNA degrades viral RNA by reads in cytoplasm as shown in the green arrow, whereas the ASO degrades viral RNA by RNAs both in the cytoplasm and nucleus as shown by the 2 yellow arrows. There are many investigational agents adding on the X open reading frame and or the S open reading frame of the virus. I will illustrate them to you in a moment. J&J 3989 has 2 RNA triggers targeting both X and S open reading frames. The study was designed to give different groups of patients with 3 monthly doses of this compound. Patients were classified into sustained responders defined by more than 1 log surface antigen reduction at day 392, that is 48 weeks after the last dose, and non-sustained responders with less than 1 log reduction at day 392. And the responders illustrated with orange lines, the median surface antigen reduction was around 2 logs. Even for study-defined non-responder, the surface antigen reduction was greater than 0.5 log at 48 weeks after the last dose. The corresponding RNA log reductions were 1.8 log for responder and 0.4 log for non-responders. These 2 figures show you the E antigen and core-related antigen reduction. Sustained responders had 1.85 logs and 1.4 logs reduction of these 2 markers respectively. A newcomer as VR2218 singly targets X open reading frame, 2 monthly injections with increasing dose resulted in a dose proportional surface antigen reduction. The surface antigen reductive effects still continue until 12 to 16 weeks after the last dose. For example, the highest mean reduction of surface antigen of more than 1.5 log was achieved in patients receiving 200 milligram at 16 weeks after the last dose. These individual scatter plots demonstrate clearly the dose relationship with maximal surface antigen suppression and with similar reductions achieved in E negative and E positive patients. Just presented in ASLD 2 to 3 weeks ago, AB729 and sRNA targeting the X open reading frame given at single dose resulted in significant surface antigen decline with the dose of 60 milligram on the left, 90 milligram in the middle, and 180 milligram on the right. Mean declines of approximately 1 log at week 12 was observed and similar between 3 groups or 3 doses. A prolonged plateau in response is observed beginning at week 12 for most of the subjects. Repeated dosing for 6 monthly doses of AB729 was associated with continuous surface antigen decline until the last point of follow-up, as shown in the left figure. In contrast, as shown by the dotted lines in the right figure, surface antigen started to return after the single dose of AB729, whereas, as shown by the red line, surface antigen continues to decline until the last assessment time point at week 20 in patients receiving multiple doses. Another new sRNA, RG6346, which singly targets the surface open reading frame, also showed very promising surface antigen reductive effect. The left figure shows the log reduction of surface antigen after giving 4 monthly injections with 3 different dosages. The right figures shows the surface antigen change in absolute values. Patients receiving 4 monthly injections with 3 milligram per kilogram would have approximate 1.8 log reduction of surface antigen. In fact, with the available longer follow-up time, as illustrated by the blue line, we observed the effect still maintained 1 year after the last dose. This slide shows the individual and mean maximal reduction in surface antigen after receiving either single dose or multiple dose of RG6346. The yellow dots represent E negative and purple E positive patients. The dots with plus sign represent patients achieving surface antigen less than 100 IU per ml, including 1 treatment-naive patients in cohort B receiving single dose. There were several others receiving multiple dose having more than 2 log reduction of the surface antigen. In addition, you can see that the responses were independent of the E status. This slide shows a single representative self-resolving ALT flare in a treatment-naive patients associated with the concomitant reduction of the surface antigen and the DNA levels and other biomarkers. The ALT elevation temporarily followed the reduction of the surface antigen and HBV DNA, suggestive of treatment-induced immune-mediated response. In this group, Group B, treatment-naive patients, 3 out of 6 patients exhibited self-limiting protocol-defined ALT flares. However, the liver synthetic and excretory functions were all preserved in these patients. In the LUT-suppressed patient receiving 4 monthly doses, there were no protocol-defined ALT flares reported. My last illustration of RNA inhibition is the ASO, antisense oligonucleotide, GSK836 given with 6 doses within 4 weeks resulted in a very profound surface antigen reduction. In treatment-naive population, the mean reduction was 1.6 log, and in nucleoside-experienced patients, it was 2.5 logs. 4 patients from each group, from both groups, had undetectable surface antigen shortly after the last dose. 6 patients from both groups of 16 patients had greater than 3 log surface antigen reduction. 3 patients in the nucleoside animal therapy cohort had ALT flares. The time course for each patient is shown on the top figures, with the surface antigen in red and ALT in blue. A temporal pattern was observed, with ALT flares initiating as the surface antigen was depleted. A similar pattern of ALT flare following surface antigen reduction due to GSK 836 treatment was also seen in a treatment-naive cohort. An example is shown here in the bottom figure. Here come to the surface antigen release inhibitor. Without bothering you the details of the coming 2 studies, which are quite complex, I just want to mention that treatment with tenofovir plus pegylated interferons plus these nucleic acid polymers with more than 48 weeks would bring 24 out of 40 patients achieving functional cure at the end of therapy. And this was maintained in 14 patients 48 weeks after off therapy. With further follow-up and combining more cohorts, functional cure was observed in 18 out of 52 patients. Using a 10-fold more sensitive surface antigen assay, all these 18 patients still have undetectable surface antigen, and they were all having unquantifiable HBV RNA and core-related antigen. So ladies and gentlemen, I would like to conclude by saying that there are many upcoming novel agents with different modes of action have established their antiviral efficacy and initial safety profiles. For viral entry inhibitor, initial positive results on the surface area reduction when combined with pegylated interferon were observed, but we don't see any additive effect or any effect of surface antigen when it is combined with nucleoside analogs. Further studies are required to confirm this surface antigen reductive effects. For RNA inhibition, either siRNA or ASO, they all had profound suppression of epigenomic RNA, surface antigen, and other antigens. We also observed a sustained surface antigen response, but we need to understand the dose and the frequency of administration in the near future. ALT elevation followed surface antigen reduction during therapy was observed and more profound in treatment-naive population. For nucleic acid polymers, initial promising results in S loss at the end of therapy and maintained in the good number of patients and longer follow-up was also observed. We also need further confirmation studies to document this mode of action with this promising functional cure rate. Thank you for your attention.
Thank you so much, MF. That was a fantastic overview. You managed to cover so much very clearly in a short period of time. So there's some very exciting results you were showing there, particularly as you pointed out for the siRNA approaches with such prolonged suppression of S antigen after just a couple of doses. And you were showing that there was often an ALT flare, suggesting that there may be an immune mechanism to that prolonged reduction. Do you know, first of all, was there a correlation between the degree of flare and the duration of suppression of S antigen?
Oh, thank you, Mela. It's a very good question. In fact, we don't have enough data to correlate the degree of flares with the duration of sustained response. But I think if it is really due to the immune awakening after the reduction of surface antigen, the bigger the flares, I would expect this, I mean, sustained response will be longer. So this is a logical thinking. If we believe this is really, the sustained effect is really due to the immune control on the virus. People are arguing whether this is really the case because we don't see many patients with flares. There are only some patients with flares and some patients do not have flares. And even some patients who have big drop in the surface antigen, they don't have flares. So we are still at a very early stage to observe this, but I mean, we don't have a definite conclusion yet, according to the results that we have for this early studies.
Great. Yeah, I guess it would be particularly the duration of the suppression that I would expect to correlate with the ALT flare, or ideally with some immunological studies actually looking at immune reconstitution, which I hope are being done as sub-studies. So thank you very much. We'll come back to more questions about all these agents in the final session, but now I'd like to move on to introduce our next speaker. So we're moving on to the immunological side of new agents, and to cover immune therapies, we have Dr. Adam Gehring, who is the biology lead at the Toronto Centre for Liver Disease, where he moved in 2016, and he leads an immunology lab there concentrating on the immunopathogenesis of hepatitis B and looking at mechanisms of actions of new therapies. So I'm very pleased to now introduce Adam Gehring. Thank you.
Good day, everyone, and I'm happy to present an update on immunomodulatory therapy for chronic hepatitis B from the AASLD Digital Liver Meeting. So these are my disclosures, and I'd like to start off with a brief rationale for immunological targets. So to date, elimination of HBV infection has only been achieved by the immune system, and a successful immune response has been well characterized in the resolution of acute HBV infection. In terms of innate immunity, we know specific cytokines like interferon alpha, IL-12, and IL-18 can have an antiviral effect. For adaptive immunity, it's a coordinated, robust CD4 and CD8 T cell response and B cell response. The CD4 T cells are required for an effective CD8 T cell differentiation. These CD8 T cells then produce interferon gamma, which non-cytolytically leads to HBV reduction in HBV replication. And CD8 T cells also eliminate infected hepatocytes, while the antibodies produced by B cells clear circulating virus and antigen. So the objective of immunotherapy for chronic hepatitis B has been to restore the level of immune function observed during resolution of acute infection. But are the requirements for HBV control the same during chronic infection? So we know the starting point to stimulate HBV immunity is vastly different in chronic hepatitis B patients. The innate immune system changes dynamically with the inflammatory environment, and this has likely led to some of the confusion observed in the literature. The literature has clearly shown that recruitment and differentiation of myeloid cells during liver inflammation, in addition to suppression of innate lymphocyte function. And this is really a consequence of the heterogeneous patient population. In terms of the adaptive immune response, it has been well studied and well characterized that there's a profound exhaustion of HBV-specific T cells. This results in a narrowing of the T cell receptor repertoire, as well as impaired antiviral function. And recent data also indicates that B cells are also sensitive to this exhaustion, resulting in impaired anti-HBs antibody production. So I want to jump right in then and start with innate immunomodulation. So there was only one study presented at AASLD looking at innate immunomodulation, and this was the TLR8 agonist from Gilead, selgantolimod. This was a phase 2 randomized double-blind placebo-controlled study where patients were viremic upon enrollment and then started either tenofovir alafenamide alone or in combination with selgantolimod at 1.5 and 3 milligrams per kilogram. So I'm not going to show the patient characteristics, but I do want to cover the ALT elevations because this is a primary concern with any immunotherapy. And so ultimately, there was an equal, essentially 15% of patients in each group experienced ALT elevations. They were slightly higher with selgantolimod combinations in both groups, between 2.5 and 5 times upper limit of normal. There was one patient with greater than 5 times upper limit normal ALT elevation, but this patient was enrolled with the 350 ALTs, an ALT of 350. So looking at HBs antigen reduction after selgantolimod treatment you can see that either the 1.5 or the 3 milligram dose of selgantelimod did lead to some HBs antigen reduction, but this was not dramatically more than the placebo alone. And this data was somewhat mirrored in the HBV DNA levels, where the combination therapy was not significantly different from that observed with tenofovir alafenamide alone. So there was another poster presented by Gilead to demonstrate that these drugs are in fact working in chronic hepatitis B patients. And in this study, they compared both the patients that were virally suppressed at enrollment and those that were viremic, and looked at cytokines in the peripheral blood. So here, this is the 1.5 milligram is in green and the 3 milligram is in blue. And what you can see is that at each dose, the patients are responding, and this response does not decrease with time, and it's not different between the virally suppressed and the viremic patients. And this is true for IL-12, IL-1RA, as well as the antiviral cytokine interferon gamma. So, we have to start to understand where the disconnect is in this translation, because now, we've seen two drugs come through phase 3 trials, one targeting TLR7, which induces interferon alpha, and another one now targeting TLR8, which induces interferon gamma. Both have shown good efficacy in animal models, and both have been validated in terms of target engagement with their immune biomarkers measured in the periphery. So interferon-stimulated genes for TLR7, and as I just showed you, IL-12 and interferon gamma for TLR8. So I think there's some important questions that we have to address here. Is one, how much of this is actually getting to the liver? Because these are orally delivered drugs. They're going through the intestinal tract, through the portal vein, where they can finally get to the liver to engage either dendritic cells or monocytes and macrophages. And I think it's going to be important to understand how much of that effect is observed in the liver versus the gut. And then a second question that hasn't really been investigated is, really, does non-cytolytic HBV clearance play a smaller role in humans compared to the animal models where these drugs have shown very good efficacy? So then, I want to move on to modulating the T cell response, and in this case, looking at therapeutic vaccination. And there was one study presented at ASLD looking at 18-month follow-up to vaccination, and this is the NASVAC study. This is a vaccine that's comprised of recombinant HBs and HB core antigens delivered intranasally as 10 doses every 2 weeks. And they tested patients that were on nucleoside analog treatment and patients that were not on treatment. And so this is the data in HBs decline. On the left are patients that were on nuke treatment, and the patients without nuke treatment on the right. And overall, the HBs decline was relatively modest at the 18-week follow-up, with 25% or 27% decline. 2 patients in the nuke treated group had lost HBs antigen, whereas 4 patients that were not on nuke treatment had lost S antigen by the end of the 18-week follow-up. They also looked at anti-HBs in the serum of these patients and found induction of anti-HBs in both groups. The nuke-treated groups started with 3% of the patients at enrollment being anti-HBs positive, with 31% of those patients positive at the end. Whereas in the patients without nucleoside analog treatment, 21% were positive at enrollment. And then after 18-month follow-up, 57% had anti-HBs antibodies in their serum. So, overall, the HBs decline was quite low in this group. There was no placebo arm to compare to, and it wasn't clear if the patients that lost HBs antigen were positive at enrollment or became anti-HBs during the vaccination. So there was another therapeutic vaccine presented as a poster from Virion Therapeutics. This is a preclinical vaccine comprised of the N-terminus of the HBV polymerase that's fused to a herpes simplex virus, glycoprotein D. They deliver this vaccine as an adenoviral vector, and the glycoprotein D, in this case, acts as a checkpoint inhibitor. And they tested immunogenicity in the AAV 1.3 HBV mouse model. And so the blue here is in naive mice, showing T cell induction in naive mice. The red is showing T cell induction in the HBV mice. And this induction of T cell immunity in the HBV model was showed reduction of viral load in the AAV HBV mouse. They also demonstrated the induction of polymerase-specific CD8 T cells in the liver of HBV mice. Further studies looked at the diversity of response to the vaccination. And you can see the diversity here, the breadth of the response in the naive mice compared to the low viral load AAV HBV mice showed a similar amount of diversity in terms of vaccine-induced T cells. Whereas if the amount of AAV HBV was increased, this T cell response started to be blunted, but was still to some extent diverse. So some of the caveats of this is that there was no control for the glycoprotein D to understand its checkpoint inhibitor effect. And then I would also say to some extent the effectiveness of polymerase-specific CD8 T cells remains unclear in humans. So then moving on to checkpoint inhibitors, so we all know that there's no shortage of suppressive factors in the liver, but it's really this PD-1, PD-L1 interaction that's being targeted as therapy and cautiously moving towards treatment for chronic hepatitis B. And we're also all aware of this pioneering study from Gilead and Ed Gain looking at nivolumab in chronic hepatitis B patients. And this one patient now has been well characterized that lost HBs antigen during this study. And some of the earlier data that was presented regarding this patient looked at the magnitude of this T cell response. And this patient that lost S antigen was presented here in red dots. And there was nothing remarkable about the magnitude of the T cell response in this patient. And I would say that's where this heterogeneity of response and not understanding what biomarkers lead to this HBs antigen reduction in PD-1 blocking has led to a lot of translational research that was presented at ASLD trying to improve patient selection. And so this is a study from the group of Carlo Ferrari looking at how the level of exhaustion relates to checkpoint inhibitor therapy. They use flow cytometry data to compare CD127 and PD-1 expression on core, HBV core specific CD8 T cells to establish an exhaustion index. And so low exhaustion index are green and blue, indicated by high CD127 expression, and highly exhausted T cells are in red, indicated by high PD-1 and low CD127. And so as controls in this study, they used patients that had achieved functional cure, either spontaneously or on new treatment. And you can see the majority of the HBV core specific CD8 T cells show a low exhaustion index. And then in HBs positive, in chronic hepatitis B patients with HBs antigen, they found that there was two groups of patients here, one with a low exhaustion index in the gray highlight, and then patients with a high exhaustion index. And when they compared this exhaustion index to the functional profile of of these cells, you can see that as the exhaustion index goes down, the functional profile goes up. And when they tested these low exhaustion index patients with PD-L1 blocking with an antibody or small molecule, these patients with low exhaustion index showed the best response to anti-PD-1 blocking. So this suggests that phenotypic markers may improve patient selection. So HBV specific CD8 T cell analysis in this really presents a challenge ex vivo, but it may allow for the potential to generalize biomarkers for patient selection. So there was also a follow-up study from Gilead looking at these patients that had a significant S decline during the nivolumab treatment. Here, they looked at 3 time points along the course of treatment. So baseline, week 12 and week 24 in 4 patients. So 2 that had HBs reduction and 2 that did not respond to nivolumab treatment. And they performed single-cell RNA sequencing on these patients and clustered the T cells and then looked for the distribution of the prevalent T cell clones using the TCR sequencing approach. And seen here in this top row is responder 1, looking at the baseline week 12 and week 24 time point. And you can see that the first 2 time points, the prevalent T cell clones are overlapping, but at this 3rd time point, there seems to be an expansion of unique T cell clones. And when you look across the other responder or in the non-responders, you don't see this change in the profile or the clones of T cells. So, you know, at this point, it's difficult to understand what this means. It's a N of 1. This could be an expansion of subdominant clones, but it may indicate it may indicate that there is unique T-cell expansion in response to PD-L1 or PD-1 blocking. And then lastly, on the checkpoint inhibitors, there was a study to look at trying to improve the timing of delivery in combination with therapeutic vaccines. So this was from Janssen Pharmaceutical or Janssen Infectious Disease. I presented this poster using therapeutic vaccination in the AAV HBV mouse model. And you can see vaccination was delivered at day 28 and day 49, and each of the colored dots are the timing of checkpoint blockade in relation to the therapeutic vaccine. And then they looked at T-cell expansion in these mice in the spleen and in the liver. And in both cases, the therapeutic vaccine alone is group 1. And overall, you can see that the blockade of response appeared better in the liver compared to the spleen. And the investigators really suggest that blocking after the boost is best. So later after the second boost of the therapeutic vaccine. But I say overall, the impact is quite small. And one important point was that blockade prior to vaccination was not actually tested in this study. And the last thing I want to talk about is a novel T-cell receptor-based therapy that was presented by Immunological at AASLD. And these are ImmTAV molecules. So ImmTAV is immune mobilizing monoclonal T-cell receptors against the virus. And these are currently in phase 1 for chronic hepatitis B. These are soluble bifunctional T-cell receptors. And these ImmTAV molecules differ from a natural T-cell receptor because they're, one, not membrane bound. They do not associate with the CD3 and instead have an effector domain that actually engages CD3 on polyclonal T cells. So these receptors remain specific for the MHC peptide presentation and engage polyclonal T cells by binding CD3 on the T cells. And so they recently published the preclinical data for this and show that, in fact, you can other antigens using this approach, and that these molecules are specific for HBV infected and integrated cells, and they are not impacted by inhibitory receptor expression because they're using a polyclonal T cell population and not HBV specific T cells. And so this is just one piece of data from the recently published paper, and this is the HepG2 NTCP in vitro infection model. And so when HepG2s are infected with HBV, you see viral replication. When you just add the T cells to this co-culture, there's no impact on viral replication. But when the ImmTAV molecules are titrated in, there's now a significant reduction in HBV replication. So one limitation, of course, of T-cell receptor-based therapies is always HLA restriction, and an interesting concept and Immunological also presented during the meeting was the use of HLA-E, which is not. So that could be a potential way to generalize T-cell receptor therapy in the chronic hepatitis B patient population. So with that, I would just like to finish and give a brief summary and perspective. So the therapeutic vaccines, so it's clear that major obstacles still remain for immunogenicity in chronic hepatitis B patients. It's unlikely to be successful as a monotherapy, and it's likely going to require combination. And there's interesting strategies that are becoming available, such as antigen reduction, combination with checkpoint inhibitors, and I think also a potential role for innate immunomodulation. Monotherapy. I say a good thing is that they have demonstrated safety, and so I think this does justify combination with additional immune targeting drugs where you can make the liver environment amenable to an immune response. And then in terms of checkpoint inhibitors, these remain promising, but patient heterogeneity is a significant issue. And you can see from the translational research that was presented during AS
Thank you, Adam. That was a terrific overview. It's clear that we all believe that the immunological therapeutics are so vital to achieving cure, and yet, set lots of obstacles that I think you've presented to us. But it was a really wonderful overview. I just want to remind everyone that if you have questions, to submit them to the Q&A. We will be addressing them at the end of the workshop. We'll have all of the faculty. Adam, I want to follow up on this issue of the exhaustion index. Sort of, for me, something that I think is a way to sort of phenotype patients in a way that might help us to select the drugs going forward. But I do want to ask you if you really think it'll be useful in the sense that, do we have any sense about, in a population of chronic Hep B patients, of this index, and how many of the patients have any sense about that? And do you think something like this exhaustion index is really going to be helpful for us going forward?
Yeah, I mean, I think it's an important proof of concept that it is possible to pull out patients that are likely to respond to checkpoint inhibitor therapy, whether and really gets to the idea that identifying assays that can do this directly ex vivo and showing that it is possible that there is that we can start to use or potentially develop that we can look at and select patients for therapy. How many of those patients, are going to be potential to respond? I think this is still quite an open question, because we look in the literature, most of these patients have been really those patients that have inactive carriers with sort of low viral load. We don't really know across the different cohorts of chronic hepatitis B patients how many of those are going to respond, and will they respond to the same? I think that part is still left to be investigated. But again, these studies looking at proof of concept immunological biomarkers and integrating these into the new studies that are coming down the pipeline for checkpoint inhibitors, it's going to be really critical, because it's likely not just an issue of magnitude of the T cell response, and we have to look at something with a little more granularity to select those patients.
Great. I'm sure we'll have more discussion about the checkpoint inhibitors and other in the question and answer period at the end of the workshop. So thanks very much, Adam. That was really very detailed and very sort of setting the stage for the questions later. My pleasure now to introduce the next speaker, Dr. Robert Gish, who is a professor of medicine at Loma Linda University, and also is the director of the Hepatitis B Foundation, and has had many titles over the years, well known to all of us, and really has been both an advocate, I think, primarily, and also an investigator in many of the studies related to hepatitis B over the last several decades. So very delighted to have Bob join us today.
Greetings. My name is Dr. Robert Gish. I'm here to present this virology meeting, Hepatitis B Cure, on Delta hepatitis, new and potential antiviral. My disclosures are on this slide. We're going to today review the overview of the hepatitis Delta life cycle. We're going to highlight preclinical and clinical targets for antiviral development that are currently in progress. We're going to also look at very recent data on result of agents in clinical development. Delta virus is always a co-infection with hepatitis B, although there's been some recent research that hepatitis Delta can also coexist, co-replicate with RNA viruses. In this setting, Delta requires surface antigen to complete virus assembly. This is an RNA virus that makes a small and large Delta antigen and has a prenylation group. There are over 15 million individuals worldwide, and between 70,000 and 150,000 in the United States. There's no FDA-approved therapy. Delta virus is the most severe form of viral hepatitis. Approximately 50% of hepatitis Delta infected patients have cirrhosis at diagnosis. This is very important to know in terms of urgency of drug development and also the patient populations that need to be studied. There's a higher risk, of course, of liver cancer, death, liver transplant, and end-stage liver disease. There are multiple potential preclinical targets for Delta hepatitis. We'll talk about the clinical ones in just a moment. But look at this wonderful diagram. Important is used for nuclear import and export. RNA transcription takes place with a number of different host enzymes. Prenylation, a host enzyme system for packaging, also is present, as well as a number of other enzymatic steps. Packaging and export also takes place, and the Delta antigen has over 100 interaction factors. Intracellular transport has also been identified. First, let's talk about Delta entry. Delta, like hepatitis B, uses the NTCP receptor for cellular uptake. Prenylation, a cell-based liver enzyme, is required for HDV assembly as prenylation takes place of the large Delta antigen using the farnesyl transferase. Let's now talk about therapies in advanced clinical development. This diagram highlights bulevirtide, an entry inhibitor, prenylation inhibitors, specifically lonafarnib, nucleic acid polymers for inhibiting packaging and release, an immune modulator, pegylated interferon lambda, that attaches to the lambda interferon receptor and stimulates downstream JAK, STAT, and interferon induction pathways. Let's now talk about an entry inhibitor. Merclid-XB, now called bulevirtide, is a subcutaneous medication that's approved for the treatment of compensated Delta in adults in Russia since 2019 and in Europe under the name Hepcludex since July 30th, 2020. This is a lipoprotein modified pre-S substance that targets the NTCP receptor. This blocks the receptor function and hepatitis B and Delta virus entry. This requires daily subcutaneous injections for 48 weeks after reconstituting lyophilized powder. 48 weeks of a higher dose, 10 milligrams of bulevirtide, as monotherapy with PEG interferon alpha-2A, patients co-infected was looked at in the MER 203 study, specifically the extension study, which we're going to be looking at today. Key inclusion criteria are highlighted here. The primary endpoint of undetectable HDV RNA at week 72, off therapy 24 weeks, was identified. Secondary endpoints include undetectable Delta RNA serum at week 48, ALT normalization, and S antigen decline. Here in the upper graphs, we're showing the representation of the median Delta RNA reduction. In green is the bulevirtide plus PEG interferon, the gray line PEG interferon monotherapy. In the right graph, you're looking at viral decline on combination therapy in blue, bulevirtide 2 milligrams, and the dark green. Let's look at the primary endpoint. This was interesting in the pegylated interferon study that the higher dose, there was a lower primary endpoint reached at 6.7% of undetectable HDV RNA. Interferon reached 53% at the primary endpoint. Let's now talk about a host enzyme system farnesyltransferase, which results in prenylation of the large delta antigen. Lonafarnib, first and only oral agent in development for delta hepatitis, has been well characterized in previous studies. Most common adverse events are GI related, identified. It's an orphan designation and breakthrough therapy designation, orphan in the US and EU, and the FDA breakthrough EME prime designation. Let's look at the phase 2 data. This is 2 lonafarnib-based regimens identified for registration. So we have lonafarnib plus ritonavir boosting. This decreases the amount of exposure to lonafarnib and lessens adverse events. And lonafarnib BID boosted plus pegylated interferon alpha 2A. The composite endpoint was reached in 29% individuals on the lonafarnib. A study that's ongoing, global study currently today, is a phase 3, and the study design is highlighted here. We're looking at lonafarnib plus pegylated interferon versus monotherapy of pegylated interferon and placebo. Lonafarnib will be ritonavir boosted. The endpoints will be a comparison of the lonafarnib 2 arms with placebo. Pegylated interferon is there for comparison, contribution of effect only. Primary endpoint again, 2-log decline at the end of treatment plus ALT normalization. The secondary endpoint of histologic improvement. We will await, excited about these new results. Let's now talk about immune modulation. This is with pegylated interferon lambda, which attaches to a different receptor than standard alpha 2A and alpha 2B pathway, interferon signaling gene induction and antiviral activity. Lambda interferon is a well-tolerated type 3 interferon, highly designation in the US and EU and an FDA breakthrough therapy designation. The LIMIT mono phase 2 study looking at lambda interferon 120 versus 180 micrograms q week for 48 weeks with a 24-week follow-up has been presented. There are subcutaneous injections, as you know. Inclusion criteria were standard with compensated liver disease. Tenofovir or entecavir was started at the baseline. Phase 2, we had a 36 ER. So interesting terminology, durable virologic response or maintained virologic response. We're not using the word SVR because that's been applied to hepatitis C, which is curable. We're not sure that delta is curable. And as long as the hepatocytes are making S antigen, there may be tiny remnants of delta infection present. This is an impressive result, with 5 out of 14 being below level of quantification at 72 weeks. Regression of liver fibrosis following 48 weeks of lambda and delta is highlighted here. These are 2 case studies with pre-treatment biopsy F5, moving to an F1, case 2, F4 with bridging fibrosis, moving to mild portal fibrosis F1 after 48 weeks of lambda interferon treatment. This was presented at the AASLD late breaker poster. Let's now talk about the LIFT phase 2 lonafarnib combo study design. This is 24 weeks, followed by 24 weeks of follow-up. Lambda, lonafarnib, ritonavir boosting with a primary endpoint of greater than 2-log HDV RNA reduction at the end of treatment, histology at follow-up. This study was presented at AASLD. Let's talk about the primary endpoint at week 24. 22 of 26 patients completed 24 weeks of treatment with 7 First, at that 48 weeks, 5 of 22, that's 23% of patients maintained below level of quantification or undetectable HDV RNA. 20 patients underwent liver biopsy, and 11 of 20, 55%, demonstrated improvement in the HAI score, and 6 of 20, or 30%, achieved the secondary endpoint with a 2-point improvement, greater than 2-point improvement in HAI. These are encouraging results. Or an interferon regimen using lambda interferon, relatively mild side effects, and GI from the lonafarnib. We'd like to highlight here hyperbilirubinemia was seen in 19%, with 4 patients undergoing discontinuation. Exploration of why these patients develop hyperbilirubinemia, including monotherapy study that I discussed earlier, is ongoing. I postulate there may be specific polymorphisms that result in this hyperbilirubinemia in certain ethnic or genetic subgroups. Integrated data for drug development includes the possibility of lonafarnib monotherapy, of course ritonavir boosted, combination lonafarnib plus lambda interferon, or possibly lambda interferon as monotherapy. We could get to this maintain or DVR, I'm going to put cure in quotations, of course, and even the possibility of maintenance therapies for this extremely serious disease. Let's now talk about that next step, which is viral packaging and release, nucleic acid polymers, including Rep2139. These are oligonucleotides with a broad spectrum of in vitro antiviral activities that appear to block both uptake or entry in some models and release in others. Rep2139 developed to address tolerability issues and is modified so you can move from an intravenous to a subcutaneous dosing. Rep2139 calcium here plus pegylated interferon was presented in 2017 to suppress HBV RNA multiple logs during mono and combination therapy. The Rep301 long-term follow-up from the Rep301 study is discussed here, with 11 301 participants completing the therapy and having 3.5 years of follow-up. 7 of 11 had this functional cure, but we're going to call that a maintained virologic response with HBV RNA not detected and normal ALT. 3 of 7 had virologic control of hepatitis B, interestingly, and 4 of 7 had surface antigen loss with a functional cure. There was a transaminase flare that took place as S antigen levels fall below 1 international units per ml. Other exploratory endpoints included S antigen with a super-sensitive assay of less than 0.005. Pregenomic RNA was quantified, and correlated antigen was quantified. Takeaway points: Delta infection infects over 12 million people worldwide. It could be as high as 20 million. It's a more severe form of chronic viral hepatitis. Interferon offers at best a 20% maintained virologic response, although with therapy lasting sometimes 5 years, we reached a DVR that's higher than 20%, but at cost in terms of medicine and side effects. Will pegylated interferon be available next year? There are statements being made that this is being removed from the market. We now have 3 major targets of HDV in development: entry, replication, and packaging. Very importantly, many of these studies require an interferon partner. If pegylated interferon standard is removed, lambda interferon may even have more opportunities. I would expect a 40 to 60% viral clearance or 2 log reduction in delta RNA with these more advanced therapies as the results become available. I'd like to thank the organizers of virology education, this important meeting of Hepatitis B Cure, which of course is looking at Delta, and my wonderful partners who help provide this information, perform the research, of course the patients and their families, and our pharma partners.
Thank you very much, Dr. Gish, for that very clear, comprehensive coverage and update on the exciting field of Delta therapy, which is really taking off now. So please, just to remind the audience, do post your questions for any of the speakers. We're looking forward to having a comprehensive discussion with the whole panel shortly after the next talk. But just before we move on, if I could just ask one question to Bob. So out of these different approaches that you presented, which one do you think is looking the most promising to build on with combinations or which combination would you say looks the most promising moving forward?
Little bit biased, Mala. Thank you so much for that question, towards the combination of lambda and lonafarnib, because there's at least 3 different ways we can use these compounds as single or dual agents. I think they've gotten the side effect profile of lonafarnib better with ritonavir, and we've also established this 2 log reduction as an intermediate endpoint as opposed to just viral clearance. So I am excited about entry inhibitors. I think they will be used in combination potentially with some of the other drugs. I'm looking at all these 3 companies and sort of modules to start working with each other to figure out what's going to provide the best level of cure. Again, we'll call it an MVR for Delta. Great question.
Lovely, thank you. Well, I think that's a good segue on to the next talk, the final talk today, which is of course such an important area, which is really looking at how to combine drugs. Because I think we all accept that to really get progress with cure for both hepatitis B and Delta, we're going to need to bring at least 2 different combination therapies together. And so to address this vital area, we're very pleased to welcome Dr. Marion Peters, who's an emeritus professor of medicine at UCSF, and is now working for the ACTG, Hepatitis Transformative Science Group, on the development of new therapies for both hepatitis B monoinfection and hepatitis B HIV co-infection. So over to you, Marion.
Thank you very much for inviting me to give an update on novel combination strategies. These are my disclosures. I'm first going to talk about combination therapies, studies from AASLD, then stopping nukes and the role in combination therapy, and then surface antigen loss abstract on natural history. So the first study I'm going to discuss is the triple combination of J&J's 3989 RNAi capsid assembly modulator and nucleoside analog from MFUN of 12 chronic hepatitis B Asian patients, 4 of them e-antigen positive, most nucleoside analog experience, who received RNAi 3 injections of 200 milligrams on days 1, 29, and 57, a capsid assembly modulator 250 milligrams a day for 12 weeks from days 1 to 85, and nucleoside analog either entecavir or tenofovir started or continued on day 1 throughout the study. And at day 113, 2 months post RNAi dosing and 1 month after end of capsid dosing, efficacy and safety data was evaluated. This slide shows the surface antigen changes from baseline to day 113 with triple therapy. On the top left, you can see the surface antigen levels in each patient. You see the J&J CAM 6379 from day 1 to 85, the 3 injections of the RNAi, and the continued decrease in surface antigen. The bottom left shows the mean surface antigen level over time. In blue is the log change from day 1 to day 113, and in orange, the actual values mean. 12 of 12 patients had greater than 1 log drop in surface antigen from day 1. The nadir was day 85 in 1 patient and day 113 in the other 11. 10 of the 12 achieved surface antigen less than 100 after 12 weeks of triple therapy. The bottom right shows the change from day 1 to day 113, and you can see it ranged from -1 to -2.2. It was no different in e-antigen positive and negative patients. This slide shows HBV DNA in patients who had greater than 100 IUs per mil on day 1, and you can see a rapid decline over the first month of therapy, except in 1 patient who had a very high level at 7.7 logs, but this declined to 3.5 logs by day 113. The bottom left shows HBV RNA in the 9 patients with quantifiable HBV RNA at day 1, and 6 of them had levels less than lower limit of quantitation by day 29. Patients positive on day 1 for E antigen and correlated antigen had less pronounced reductions in these parameters. This triple therapy was safe without deaths or discontinuations. There were no clinically significant laboratory findings except for 5 grade 1 transient isolated ALT elevations, which resolved with continued dosing and were associated temporarily with reduction of viral parameters. In conclusion, it's the first study to evaluate the efficacy and safety of triple combination therapy, RNAi, capsid assembly modulator, and a nuke. It was well tolerated with robust reductions in surface antigen, HBV DNA, and HBV RNA. All patients achieved greater than 1 log reduction in surface antigen, nadir from -1 to -2.26. Surface antigen reductions were similar in E antigen positive and negative patients. Studies of longer duration with this triple combination are underway, aimed at assessing functional cure in patients with chronic hepatitis B. The next abstract is the timing and administration of anti-PD-1 in evaluating HBV-specific immune responses in AAV HBV mice. The study's aims were to assess whether therapeutic vaccine could increase HBV-specific T-cell responses. The vaccine to core and pol was given 28 and 49 days after HBV chronicity was established in the mice, and then to evaluate if anti-PD-1 enhanced immunogenicity. At day 63, splenocytes and intrahepatic lymphocytes were isolated, gamma interferon was assessed by ELISpot, and T-cell proliferation, ALT, HBV DNA, and surface antigen parameters were measured throughout. On the right-hand figure is the vaccination and dosing scheme. Group 1 is the vaccine alone. PD-1 is given in groups 2, 4, 6, 8, 10, and the isotype control in groups 3, 5, 7, 9, 11. These are the results from the splenocytes. The therapeutic vaccine induced gamma interferon in secreting T cells in group 1, and addition of PD-1 with or after the vaccine did not improve immunogenicity on the left, nor proliferative capacity on the right. And most mice had 100% receptor occupancy with anti-PD-1. These are the intrahepatic lymphocyte results. The therapeutic vaccine induced gamma-secreting T cells in the liver, as you can see on the left in group 1. In addition of PD-1 improved immunogenicity, but they stated that isotype control also did, but didn't show any data. Addition of anti-PD-1 improved the proliferative capacity on the right, but only in group 6, where anti-PD-1 was given a week after the booster vaccine. Receptor occupancy was not 100% in the liver, and it did not correlate with immune responses nor proliferation. So they studied HBV DNA and surface antigen. There was absolutely no change. ALT results were not given. In conclusion, they said that anti-PD-1 receptor occupancy did not correlate with immune responses, and anti-PD-1 should not be given with therapeutic vaccine or given at least 1 week after booster vaccination to see a beneficial effect. Now I want to look at studies of stopping nukes at AASLD in 2020. The first is the RETRACT-B study showing surface antigen loss is higher among Caucasians compared to Asians after stopping nucleoside analog therapy. It's the result from large global multi-ethnic cohort of 1,541 patients, mainly Asian, predominantly genotypes B and C, small number serotic. The mean quantitative surface antigen at stopping nukes was 2.6, and they had normal ALT. Surface antigen loss after discontinuation was 3% at 1 year and 14% at 4 years. Univariate analysis showed that surface antigen loss was higher amongst older patients, 18% at 4 years compared to younger, 9%. Higher on those receiving tenofovir, 17% at 4 years versus entecavir, 12%. No different between e antigen positive and negative, and higher among Caucasians, 41% at 4 years compared to 11% in Asians. Multivariate analysis showed that only the Asian Caucasian difference was statistically significant, and Caucasian had a 6 times higher likelihood of clearing than Asians. Retreatment occurred in 30% at 1 year and 56% at 4 years. 1% of patients had decompensation and a similar number died. The next study from the HBRN with Nora Tarot showed a randomized controlled trial of 196 weeks of tenofovir with or without pegylated interferon for the first 24 weeks, followed by protocolized withdrawal in adults. And on the right in the graph, you can see the percent of surface antigen loss, which increased during therapy in the tenofovir plus interferon level, but after therapy was discontinued, increased in the tenofovir level, so there was no difference at week 240. Tenofovir was stopped if patients' HBV DNA was under 1,000, they did not have cirrhosis, they were e antigen negative and anti-E positive. The table shows the surface antigen loss on those who stopped nukes and those who did not. It was 6.3% in those who stopped nukes and 2.6% in those who did not. Inactive status was higher in those who did not stop but maintained tenofovir at 64% compared to 30% who stopped. There was no benefit, they concluded, of PEG tenofovir over tenofovir alone. They noted similar flares between the two groups, but flares occurred mainly during interferon and PEG interferon and tenofovir therapy, whereas with tenofovir alone, flares occurred after withdrawal. They note that you may need longer follow-up to assess surface loss. The next study is a small study from Calgary looking at a single center experience on the efficacy of stopping nuke therapy in patients with chronic hep B. It was 1,337 patients, 47 stopped therapy on MD recommendation, which was low surface antigen and low fibrosis. 6 who restarted all had flares from 139 to 1467 ALT, and they all occurred in the first 6 months after stopping nukes. Of interest, quantitative surface was very low at the time of stopping nukes, from 2 to 205. 41 patients are still off nuke therapy and 1 has lost surface antigen so far. The last study is a surface antigen loss in community-based untreated chronic hepatitis B cohort, the REVEAL study of 4,100 patients, and they took 707 patients who did not have hepatitis C, were not cirrhotic, and at least 3 data points in the last 5 years of follow-up. 5.8 had lost surface antigen, and the vast majority had not. They did the quantitative surface antigen at baseline. In those who lost surface antigen, it was 282, whereas in those who did not lose surface antigen, it was 1196. I took the baseline to mean the first of the 3 time points. They then went on to analyze changes over time. And you can see in the box plots that in red are the patients who did not clear surface antigen and their quantitative surface did not change. Whereas in blue, they did clear, and you can see a stepwise decrease in surface antigen. And their analysis statistically said that the rapid surface antigen decline of 0.5 logs per year predicted surface loss. And that was as good as a previous analysis using 200 actual value plus 0.5 per year, and better than using a level less than 200 alone. So in summary, there were few studies on combination therapy reported at AASLD except with nuke therapy, and there are multiple combinations. The timing of the combinations are unclear. DAAs can have a profound reduction of quantitative surface, but will this lead to long-term inactive off-treatment state or even loss of surface antigen? Or will more be needed to obtain a functional cure with loss of surface antigen, such as interferon, other immune modulators, or therapeutic vaccine? And I guess one of the questions coming up from ASLD is response-guided therapy needed, or will you need to stop nukes to achieve functional cure? I don't know the answer. Thank you very much.
Thank you, Marion. That was, you've been given a hard task actually, and I'm sure in the question and answer, you're gonna actually be asked to put your money down on what the new combination should look like. But I'm gonna start by just asking you a question about the withdrawal, since that's sort of our current therapy, sort of our, there's a lot of discussion about withdrawal of nukes and sort of its role. And I frequently get asked myself, is it ready for prime time? So I'm going to ask you, you know, based on really what the data presented at ASLD, have we moved the needle? Is it shifted to a point now where at least while we're waiting for the new therapies to come, is there a role for withdrawal, and in whom would you say that strategy should be considered?
I think I added the withdrawal therapy because many of the new combination studies coming up in the future are using nuke withdrawal at the end of their DAA therapy. And since there was a lot of it, the points that came out is I don't think we really know enough yet about nuke withdrawal. Some studies say it's as high as 30%. The ASLD abstracts were in the single digits, and relapse was frequent. This Asian Caucasian difference hadn't been shown in a lot of other studies, and therefore, until we really can define the best way to assess it, I like the data that looked at quantitative surface decline over time, and I wonder if that may be one of the roles we should be using with surface antigen. And I know if you could go back in the HBRN, you probably have that data when you do the manuscript to really look at, does that predict those people who will lose surface antigen and get a functional cure? So at this time, I think we still need to do it within studies because there are too many unanswered questions.
But I think you make the excellent point that that's sort of thinking about the future. It's this movement to finite treatment. And so we put treatments on, we're going to pull them off, and sort of understanding how that should be done is really what we can learn from the withdrawal studies, potentially.
Yes.
Very good. All right, we're going to talk more about combination therapy. I'd like to invite all of the speakers back. We have some time to share questions from the audience. Thank you to those who have submitted their questions. And I would encourage you to still continue to send your questions through the Q&A. We'll try to get to as many as we can in the time that remains. So I'm going to start by giving a question. There were quite a few questions that came in about the siRNA. So MF, you're going to be on the hotspot to start. So one of them was from Dr. Zulum from Fabian. Says if the siRNA targets the 3' end of the HBV genome, this will target all viral transcripts, including HBX. My understanding is that the X transcripts are also targeted by other siRNAs from other companies. Can you just clarify exactly kind of the role of X? That was a prominent question around the siRNAs. How important is it to target X in that strategy?
Thank you very much. In fact, this is the real thing that we know, that if we can target the X open reading frame, put it this way, then all the transcript, including the S, genomic RNAs, and also the polymerase, etc., all the transcript will be locked down. Whereas if you're just targeting the S, the surface open reading frame, you will, because of the non-overlapping transcription, that you will leave the X transcript intact. So for all the companies that investigating all this siRNA, there's only one company, the Dicerna, actually used the what we call only targeting the S open reading frame. So that sRNA will leave the X transcripts intact. Whereas all the other companies, pharmaceutical companies, they use the sRNA, including the X open reading frame. So if you use that one, then all the transcript will be shut down. So this is the difference between the triggers different companies are using. So there's only, in conclusion, there's only one sRNA in the study arena now that they use only the S targeting sRNA, leaving the X. All the others targeting the X will shut down all the transcripts.
And do you have a sense of really sort of the value of shutting them all down versus being more selective? Do you think that ultimately that's going to translate into an outcome that's going to differ?
Yes. If you look at the surface antigen reduction, okay, what I can see comparing these two, different strategy, whether you leave or lock down the X, the surface antigen reduction seems to be the same. Okay, in clinical study, my study, what Rationale has shown is in my study, when you preserve the X, then the surface antigen lockdown is less compared to leaving the X intact. So clinically we don't see it, but in the mice experiment we actually find that there's a difference. And people are constantly asking if you leave the X, which is a transactivating transcript, and that may increase the risk of HCC, but we don't know. This is a theoretical concern, but we need to see. So this is the difference that we see in mice, and we don't see a difference in terms of surface antigen reduction, although we don't have a head-to-head comparison between the sRNA.
Very good, thanks. That's Mala.
Okay, before we let MF off the hook, there's another interesting question from Adrian Kuiperi. He's saying very impressive sustained reductions in S antigen with the siRNA. Can you speculate on whether you think this is due to reductions in the individual infected hepatocytes or reductions in the level of cccDNA within all hepatocytes? Do we have any insights into that?
I think we don't have an answer yet, but I think when you look at all the sustainability of this, surface antigen reductive effect, we are thinking of whether this is because of the immune control on the infected hepatocytes, shutting down the controlling the viral activities. So what we know is this sRNA won't have a direct effect on cccDNA. So if we have the immune control after reduction of the surface antigen, then you will expect to have a decrease in the infected hepatocytes subsequently. Another thing we also don't know is whether all this risk complex or even for the ASO, the RNase, what is the time of the occupancy of this sRNA or ASO intracellularly. So that may again explain partly why we may see some sustained response in terms of the surface antigen reduction.
Is anyone looking at sampling the liver in any of these studies to try to get a handle on what's actually happening?
This is a very good idea, but for the phase 1b study, we don't have the liver tissues for examining the half-life, the occupancy, even the turnover of the receptors, etc., to explain the sustainability of the effect that we saw in the trial.
Okay, thank you.
I'm going to shift over to Delta for a few minutes. Jordan Feld submitted a question, Bob, asking, given the high rate of relapse with Lambda, do you, as a monotherapy, do you think that's going to be an issue as they go into these combination trials? Can you speak to sort of the sustained responses? And then I have a follow-up question for Adam, actually, just about the role of Lambda interferon. Is there any role for it in the HBV arena? We haven't talked at all about that, and given sort of resurgence in Delta, do we see any place for it in hepatitis B in terms of combination therapies, for example?
That's a great question, Nora. And of course, each of these drugs may end up being used alone. I think in really low viral load patients with Delta, Lambda alone may be adequate, or lonafarnib boosted with ritonavir may be adequate to get a maintained virologic response. In general, I agree with you. It's not going to be good as monotherapy across general populations, and definitely not with high viral loads. And the other question with Lambda is, why is there a subgroup of these patients that get this hyperbilirubinemia without any signs of real liver dysfunction? Otherwise, albumin stays normal, there's no ascites, there's no coagulopathy. So I really think they need to do some type of genetic profiling to find out why a subgroup of patients develops this side effect.
Okay, yeah. So to follow up on, I mean, it's an interesting concept, Nora. I mean, we see interferon alpha being put in as an immunomodulator on top of on a number of new DAA trials. And so, yeah, certainly I wouldn't rule Lambda out here. I mean, its receptor expression and potentially lower side effect profile might be a good combination to include in some of these going forward. So I think it's an interesting question that we have to watch.
So can I just ask you, Adam, just in follow-up, we're going to kind of get to this idea of like, what would we, if we had the dream combo, what would it be? And I think everybody would say we'd envision an immune modulatory therapy in there, but can you kind of give us a sense of, if you were thinking about either how to use an immune modulatory therapy as part of a combination, would it be concurrent, sequential, just based on sort of what you understand about this disease? What would be sort of the strategy that you would put forth as being the one that we should be evaluating?
Yeah, I mean, it's obviously, it's a really interesting question, and that there's a lot of rationale and logic that goes behind thinking about how to combine these different drugs. I mean, the effect of the surface antigen reducing drugs is profound, the siRNAs and ASOs. And putting these in combination with something like a therapeutic vaccine makes a lot of sense. I mean, you remove that tolerogen and then you boost the immune response. But in that case, you have to eventually allow that S antigen to potentially come back to recognize infected hepatocytes in the liver. And so, for instance, giving an siRNA and ASO that might reduce S antigen in infected hepatocytes and giving a PDL, or PD-1 or PDL1 checkpoint inhibitor likely makes less sense. Right? So you would not get recognition of infected hepatocytes. In that case, maybe enhancing antigen presentation or activation in the liver. So you give an innate immunomodulator in combination with a checkpoint blockade might actually be an interesting combination. So, it really comes down to the mechanism of action for the DAAs and the combination with the immunotherapy drugs that, we can start to make sense of with some of the preclinical data, but we don't have, I mean, obviously these haven't gone through into patients yet to understand. And there is, as was pointed out for the siRNAs, some lack of information in the liver of how the different drugs are actually impacting the liver-specific immune response. So, in that case, you can only get so complex when it comes to combining these, or else the treatment becomes untenable. So I think at this point, really pairing this antigen knockdown with either potentially innate immunomodulation could work, or antigen knockdown with therapeutic vaccines, as some of these newer vaccines that are just coming into phase 1 that seem to be more immunogenic than what's been tried in the past. I think these could be relatively promising. So, it's a difficult question that comes up everywhere, and there's a number of different combinations that can be proposed. In the end, I do think that even though the innate immunomodulators haven't been successful as monotherapy, using them in these combinations to change the liver environment might be important, though.
Nora, can I follow up with a question which is very much related, which was burning?
Please.
Yeah. So Adam, and maybe also Mala, we've seen now several TLR agonists. The short question is, which is your favorite to cure HBV and why?
I mean, I can start with this. I mean, and I kind of touched on it briefly in my talk, Harry. I mean, you put these similar compounds in animal models, and they're very effective. In the woodchuck and the chimpanzees and the mice models. And so, that direct antiviral effect of these, the antiviral effect of these is just not translating into humans. So I do believe there is more we need to know to make sure that they're actually working in the liver. In terms of what we want to achieve, if we're thinking that the T cell response is important, probably the TLR8 agonist is likely to be the most effective because we know it induces IL-12, but just providing IL-12 to T cells may not be enough. Because in terms of the TLR7 and interferon alpha, and we know interferon alpha has been used for a very long time in hepatitis B, and at least systemic administration is only moderately successful. So, in that case, when we're thinking about immunotherapy, we want to think about inducing the T cell response. The TLR8 agonist is probably the combination that I would start to put into the mix.
Yeah, and I think we because of the complexity of their mode of action, particularly TLR8 agonists, which act on so many different cells, I think a better understanding of that will allow us to use them more appropriately in the future with the right combinations. They might need additional drugs to go along with them to sort of harness the good immune modulatory effects and block the pathogenic effects they're having.
Marion, did you want to make a comment?
Yes, I wanted to make a comment from the patient point of view that patients feel stable on nukes, so you have to offer something as a functional cure that's finite, short, and not torturous. And so, all these wonderful drugs that you get injected or IV or whatever, patients are going to go home, talk to their family, and call back and say sorry. So I think really Adam's point is make them finite, make them short. And already there are 4 different companies doing add-ons of interferon, which won't be super popular. But I think Harry's point of TLR8, which is an oral medication, yes, it doesn't do a lot on its own, but it doesn't do a lot bad. And that might be a perfect combination that, if it worked, would have great patient acceptance. And I think we have to keep thinking about that.
Kosh?
Can I just follow up with you, Marion,
sure.
before we come to Kosh? So while you're talking about combinations that are patient-friendly, what would be your preferred combination at the moment?
So I think the ones that are farthest along is a few RNA interference injections, plus oral CAM, plus a nuke, and then have a finite stop therapy. And then my other part of the platform would be adding a TLR8, which is easy. And then if you have some very brave, strong souls, adding interferon as an alternative. But I don't think interferon's going to have great excitement, but it may be the ribavirin of hepatitis B. We may be stuck with it.
Okay.
Kosh?
So I think it's a really interesting discussion, and I'm not sure we know that there's a clear pathway forward. I was going to tease Adam, as I always do, to say that we've not really kind of seen that translation into patients, the excitement that we want to see with these immune modulators. And that is really something that I think is going to still be a year, 18 months away until we understand a little bit more about the phase 2b studies with combination antivirals and certainly siRNAs and CAMs. I wanted to push back a little bit on Marion, because otherwise it won't be fun, to say that interferon kind of can give us a lead in, because we know what it does in patients. We've had a sort of plethora of drugs that have not really translated, as I've just said. And interferon, we know what it does, and it has some effect. Harry, Nora, others have published huge amounts on this drug. So I do understand the patient perspective. I very much agree with the patient perspective of, for example, discontinuation of nukes and a low replicative state not being an acceptable outcome from the data we saw at EASL and at ASLD. But I think that that interferon may be very short term as just a mechanism to show us what immunomodulators can do, either with a timing perspective or to just show what a generic immunomodulator can do, until we're more confident about the TLR7s, the TLR8s, and everything else. That's just my personal view.
No, I agree, Kosh, that interferon is the best we so far have for getting rid of surface antigen that's available. So I agree that has very profound immunomodulatory effects. It just has to be short to get patient acceptance. And I don't think we should forget this. We can't have a study that goes 2 years. We have to have a study with short therapy and then long follow-up, because that's the other point we haven't brought up, that surface antigen may be lost 6 months, a year later, and we need to capture those data.
That's a fair point.
Now I follow up with you, Kosh. You talked about how many different capsid inhibitors are becoming available. So did you, in reviewing the literature, did you have a feel for a group or a particular one that looks the most promising one to include in a combination going forward?
So I think this is a discussion for the panel, but I'll go first because, again, it's sort of choice and kind of what we enjoy doing. I think that I'm not so sure, and I can be corrected by cleverer people on the panel, that we fully understand the differences between the different classes and an effect that may be different, although we understand sort of the aberrant versus the empty kind of classification. I still think there's this nuance, which I think is important for the whole field, about understanding what on-treatment, quote unquote, rises in ALT are actually meaning, whether they're flares or not. I think that's relevant in reviewing the sort of capsid field. I think that everyone's got one, and I think that they are going to be a backbone. Whether we see significant clinical effects with longer term diminishment of CCC amplification with newer generation of capsids is the promise of capsids, but I think we just don't know yet. So, and then I'd open it up to my colleagues like MF and others who are very active, because they will have their views.
I totally agree, Gosh, that we still need to find more, evidence for the direction for the capsid inhibitors, because all the trial that we are running is usually relatively short, 4 weeks, and we do have, two to three longer therapy, 24 weeks of CPAM, but it seems that the effect is only on the further suppression of DNA and also RNA. So we are yet to see whether the surface antigen, the other rival antigens will have greater effect on longer therapy. But then we have sRNA already. So the field is actually quite complex in that sense. How we can actually use all this different drugs with different mode of action to enhance its functional cure.
And I think my comeback, and again, I'd open it up to Harry and Nora and yourself, Marla, and Bob, is that actually it's even more complicated, because we certainly do want to have the immunomodulators, but we have no idea when the timing of those should be. Should they be induction? Should they be consolidation? Should they be intermittent? I think it's fairly conceptual at the moment, and I think we need to give a little bit of time to let it evolve out. I'm very much in your camp, both Adam and Marla, with regard to we need more intrahepatic sampling. There are going to be a couple of studies that are very intense with regard to sampling in small groups of patients. I think they're going to be seriously tough to recruit to, but I think we do need to understand a bit more about the first generation combinations of siRNA and capsids to see what they're doing, to then think about the timing of immunomodulators. And I think that means we're going to need to be a bit more patient, I would say.
But I think, Kosh, you highlighted that CAMs are not going to be used as a monotherapy. But they're oral. They're they've got the tolerability issue, so they're very nice kind of companion drug. I sort of view nice to consider in a combination therapy. So I want to ask you, and that's actually about sort of the combos that you would sort of envision, CAM plus siRNAs, for example, et cetera. So, because looking at finite therapy, because I think one of the things that's really exciting about new HBV therapies is we're looking at finite amounts of treatment, 6 months or often even shorter, and then looking to see what we get long term. But can you put together your favorite kind of combos? So I'm going to ask you to sort of speak to that. And I'll start with you, Kosh, because I think CAMs will be part of that combo. They've got that sort of, I think, the...
It's more fun if we ask MF, because he's tired. No, you go first, MF.
This is really a difficult question. I think Marian's also presented the data of the triple. If you look at that data, in fact, the reduction of surface antigen is actually not superior to double, meaning that the nukes plus sRNA plus CPAM versus nukes plus sRNA, the surface antigen reduction is actually same as far as that particular study is concerned. So I think, I mean, if we are thinking of combo therapy, the role of capsid CPAM now is to further suppress the DNA and RNA. The role of sRNA is to suppress the surface antigen and other viral antigens. So I think, I mean, I have no difficulty to combining all three together, but whether combining all three together will actually shorten the time that we can achieve the functional cure compared to just double, I don't know.
Better surely to have the third agent being an immunomodulatory agent. And we haven't talked that much about therapeutic vaccines. I was disappointed that Marion didn't include one in her ideal regime, because I do think trying to focus the response on the antigen-specific component with the vaccine in the setting of everything else being idealized is a really good idea. So the vaccines that you presented an update on, Adam, from Basel, sounded a little bit unusual. The NASVEC, I mean, I could understand nasal targeting for COVID, but what's the logic in doing that for?
Yeah,
I mean,
yeah, I mean, to some extent, you know, I would agree with you. Plus, there was, you know, I think 10 administrations of this. It's a recombinant antigen vaccine, and they were presenting the follow-up data, and I think you saw that it wasn't particularly robust. And you know, there hasn't been a lot of public presentation of some of the therapeutic vaccine data recently. I know there are much more immunogenic ones coming down the pipeline, because we know historically the recombinant antigens have not been effective and are unlikely to be effective, even in the case of reducing the S antigen load, unless there's some kind of revolutionary adjuvant that comes along. So I really think the space for therapeutic vaccines is these vaccines with multiple components: heterologous, prime, boost, you know, adenoviral vectors. You know, this strategy is likely to be much more effective when paired with the other direct-acting antivirals. So, you know, this one, there wasn't very much presented at AASLD or even, I believe, at EASL the last time. And so, because I still think it's quite early on for a lot of these new generation of therapeutic vaccines, but there are definitely much more immunogenic ones coming down the pipeline.
So Adam, do you think the therapeutic vaccine should be given concomitantly with the DAA, or once you've driven a little bit, not maybe get rid of surface antigen, but driven it down? I mean, with that HBV mouse model suggested therapeutic vaccine could enhance intrahepatic responses. The question is, when should it be given?
Yeah, I mean, certainly the mouse model data look compelling for a driving the surface antigen down below a specific cutoff. You know, what that's going to be in patients, whether that's going to translate in patients, I mean, that's going to have to be tested. And it seems, to me at least, logical that the surface antigen should be down for a bit before you start giving the vaccination. You know, but you can see, you know, even for the study that you had mentioned with checkpoint blockade, and I also had mentioned that the timing of that is still something that really needs to be investigated. But, you know, I think it should certainly be a few months after the antigen is down, and to try and provide the best boost.
Can I ask a question to Marion, Nora?
Please.
Yeah. So Marion, you talked about stopping therapy. And the question is, should we use stopping therapy as a true entity in new regimens to get cured? So to plan stopping, like stop and shock and that type of treatments, what is your view on that?
So the short answer is yes, because if you're going to go through all of this, you want to be off therapy at the end as a functional cure. So yes, you will have to stop nukes at some point to show that you have a functional cure. And most of the companies now have invoked a stop nuke point at some point during the end of their combination of RNAi or plus CAM or plus interferon. So I think we will have to invoke stopping nukes, but will we be able to do it in everybody is a very different question. And I think part of what we learned at ASLD is we don't actually have the data to tell us exactly who to stop and when, and will it be safe in everyone. But I think if you're asking patients to go on all these fancy schmancy drugs, there has to be an end of therapy. It has to be off therapy at the end, or they'd be better off just staying where they were in the first place.
Oh, but this is, yeah. So this partly answered my question, because obviously we have to show on treatment that we're getting cured, and the only way to do that is to show off treatment sustained response. So we would have, but is there a role for stopping therapy, for instance, for interruption of nuke analogs, nucleoside analogs, and then moving on? So to really use it as a therapeutic entity within regimens. That is the question.
Sorry, I misunderstood. Yes, there's a great role for that, because it may be if the people are suppressed and inactive on nuke therapy, and you boost the immune response when you stop therapy, then coming with a finite course of DAA plus or minus immunomodulator might actually lead to a better cure.
But Marion, don't you think the challenge with that strategy is the sort of the shock part, which is, you know, they get the ALT flare, nobody really knows how to kind of predict them. And so I think that's always been the scary part about, yes, this idea that we want to see these sort of flares, we want to see these sort of, these changes that tell us that immunologically something's happening, but the inability to know, kind of to predict how they're going to evolve, kind of how severe they're going to be, that remains a challenge, I think.
But we're getting
sort of intermittent.
Yeah. We are getting more data. I mean, to me, a bad flare is one that leads to decompensation. It's not. It may still be associated with dropping surface antigen, but if it leads to a bad outcome in the patient, then that's a bad flare, whether it's toxic or not. So I think that it's important. We have data monitoring patients when they flare. We have criteria to know when to restart therapy. I mean, the HBRN came up with, I thought, very good criteria in how to do that. And if you use those criteria, you could still identify patients, even if they flare, who could be then continued on whatever therapy your second part of the platform is. So I think it's not easy and would have to be done very, very, patients followed very, very closely, but it may be a great way to induce a functional cure. Or you could say, maybe we should just be stopping therapy and not worry about DAAs. That would be bad for industry, but there are a percentage of patients who do well.
Gosh.
No, firstly, to Harry's point, I think it's an appropriate strategy to think about stopping nukes as an induction to newer therapies, particularly immunomodulatory, with maybe induction of a further different mode of action, antiviral, whatever you might want to do. I certainly think that's an interesting concept. I'm going to slightly disagree with Nora. I think we need to move the dial a bit, and we need to think about being comfortable about managing abnormal liver function, as in hepatitis flares in hepatitis B, and being more comfortable with it. It's going to be difficult. It's going to need a lot of education, but that's the reality of what's happening in some of the trials now. So certainly in the REEF-2 trial, it's 48 weeks in non-cirrhotic patients, an siRNA and the J&J capsid, and it's stopped. And then let's see how things go and let's manage them. How we support all the PIs and what we will learn around that, I think, will be extremely informative for the field. Of course, it'd be better if we were doing some more intrahepatic measurements. Marla, Adam, I agree. But I think we've got to be more comfortable, and it may be that we throw a lot more at patients in a shorter time period to try and make sure that it is finite. Otherwise, it's going to be a long road.
No,
Can I ask a question?
Sorry, please, go ahead.
Thanks, Nora. About the Altimmune product, which is called Hep T cell, which has got 9 fluoropeptides derived from the hepatitis B virus. I wanted to see if people had comments on this idea. It's going into a phase 2 study right now. Adam, if you might have comments on the possibility of that redirecting T cell activity and immunity against hepatitis B?
Yeah, so I'm certainly aware of the Altimmune data. I mean, they presented some, I think it's quite some time ago now, in their earlier studies showing some immunogenicity, not a tremendous amount. I mean, peptide vaccines in general have been also challenging in chronic hepatitis B. You know, they are predicted epitopes, I believe, in their synthetic peptides, so they may have, and they do have a novel combination. A novel conjugation that I think allows them to be more immunogenic than past peptide therapeutic vaccines that have been tested in hepatitis B. And you know, these all come down to, is that vaccine going to be a good enough match across the different genotypes of HBV? You know, what is their novel conjugation enough of an adjuvant to really boost that? And you know, obviously they believe so. They're moving into this phase 2 study, and you know, like with most therapeutic vaccines, you know, with the strategy right now showing that you can induce vaccine-specific T cell immunity is important, and it's important to get into that combination strategies that are moving forward that we're talking about. So I think that's really the goal. Would be at least in my perspective here, in trying to move these different therapeutic vaccines forward, including the one from Altimmune.
Can I just quickly ask you, Bob, is anyone developing therapeutic vaccines for Delta? Because with the lack of sustained response that we're seeing, even with these exciting new agents, trying to harness a specific adaptive immune response would be an interesting strategy to come in with at the end. Yeah.
Great question. I've kept my ear to the ground and haven't heard anything about that idea. So I'll have to bring that back to the HDIN group. That's a great idea. Can I make one more comment from an advocacy perspective? I want to put on that hepatitis B Foundation hat. I really think from a terminology perspective, if we keep liver enzymes separate from liver function, will be really important to patients, because if you tell them their liver function tests are off, they get confused, they get concerned. And back to the flare issue, if it's just enzymes, I think Marion was talking about, well, if the bilirubin's fine, then it's a good flare, well, provided you have a virologic response. So we could help our patients with that terminology, it'd be quite reassuring.
Can I make a point? I think it's a really important point you make, is how we manage the terminology of flares, and that we remember, and some of us on this panel have enjoyed that sometimes seeing a rise in AST and ALT, if it's accompanying a surface antigen decline, might not be a good flare. So there's still quite a lot of safety aspects that we need to think about, and understanding how the patients feel about this and reassuring them, I think is critical. But I think it is doable, and I think we will be able to do it with the right advocacy and the right presentation of the aims of various studies we're doing.
I'd like to make one last point, kind of follow up on this flare issue as well from caution. It was discussed earlier. You know, really, you know, in terms of the combinations with immunomodulators, whether it's T cell or innate, understanding the profile and the pattern of those flares and how they're happening with these direct acting antivirals is critical before you start thinking about doing any kind of immunomodulation, because you don't want to tip the needle too far in the wrong direction. So really, you know, it's important looking closely at the data from these phase 2 studies and really understanding the profile of what those flares look like with the DAAs, and you know, their duration, their intensity, their association with S decline. You know, these are, you know, once this pattern is really understood, then, you know, combining them with the immunomodulator then, you know, may become more feasible, or may not be recommended.
Okay, well, we're now just running out of time, so I think we won't be able to have any more questions, but it's been a fantastic discussion. Thank you all for your participation, and thank you for the fantastic talks. And I'd just like to wrap up the session on behalf of Nora and myself by handing back to Harry to close up.
Yeah, thanks so much, Mala. I'm happy to close this meeting. It's been wonderful. This is the seventh time that we've done this. We started with these international workshop on HBV Cure in 2014, and unfortunately we had to do it online this time, but that didn't make it really less effective. And I really want to thank the speakers and the chairs. It was really a world-class faculty today that we had, and that was also, I think, illustrated in the discussion, which was quite high level. I also like to thank the attendees who will be there, and I would like to thank Virology Education. And I have a few remarks, I think, which are in the slides, right? Can you forward them? Yeah. So there will be a certificate of attendance for everyone, but you only get that once you have the meeting survey been sent out. So I hope you will be able to do that. It will give us much more indication on how we can tweak the meeting for next year, which will hopefully be in Toronto again, live to meet people in person. That would be wonderful, and we hope we'll get by this COVID pandemic at that time. Next slide. So all of this will be available online via the On-Air by Events Air virtual portal very soon. And the login code is your registered email address and your personal PIN code, which will be sent to you separately. Next slide. I'd like to thank all the industrial partners who supported this meeting. Thank you very much. Without you, we would not be able to do this. Thank you also actually for investing a lot of dollars and efforts in getting this hepatitis B disease cured, which we're all involved in, and it's an extremely exciting era now that we can get this nut to crack and really get this disease cured finally. Next slide. So I'd like to thank everyone for joining us. I really hope to see you again next year in Toronto. And I wish you all a very good morning, afternoon, evening, wherever you are. Thanks so much.